Okay, this is the third video on, we're going to look at seed dispersal, dormancy, and germination. Okay, at this stage, the plant has formed a seed from the ovule, and there's a fruit surrounding the seed, which is made from the ovary. Okay, the seeds need to be dispersed. They need to get away from their parent plant. If the seeds were just to fall directly onto the ground, they'd be in the shade of the parent plant. They wouldn't be able to get enough water from the soil, so they need to get away from the parent plant to avoid competition. They're avoiding competition for space, light, and water. Even getting away a few meters is enough. Okay, the seeds are surrounded by fruits. The fruits are structures specifically designed to help dispersal, to help the seeds get away from their parent plant. The fruits can be dry, like a sycamore seed, or they can be soft and fleshy, like a tomato. You're required to know that seeds can be dispersed by four main methods. Okay, we have wind, water, animal, and self. And you should know an example of each. So in wind dispersal, the plants make use of the wind to blow the seeds some distance away from the parent plant. So the fruit is going to be modified in some way to catch the wind. So the example you might learn with sycamore, and seeds are surrounded by a fruit that are modified as wings. So we have something similar to this. We'd have wing-shaped fruits and a seed at the center. Other examples will be dandelion. Dandelion seeds have little parachutes that are caught by the wind and carried many kilometers away. So the wind can scatter the seeds. Water, many plants produce seeds that can float, and if they're beside a stream, the seeds will be carried away some distance. So water lily would be an example there. In animal dispersal, two possibilities. The fruit and seeds can be eaten by the animal and then digested some distance away, or the seeds can have hooks on them that attach to the animal's fur. And again, they can fall off some kilometers away. So example of uh, animal dispersed seeds. Okay, the tomatoes eaten by animals, and the seeds pass through the animal's digestive system and are egested somewhere else. Okay, seeds like thistles, the fruits have little hooks on them that can attach to an animal's fur. Whatever works to carry the seed away from the parent plant. Some seeds can actually self-disperse, that they have a mechanism that as the fruit dries, it twists and a pressure builds up and it ends up flicking the seeds some distance away. These are the examples here. If you take the pea pod as an example, as the pea pod dries out, it twists, the pressure builds up, and suddenly it flicks the seeds a short distance away from the parent plant. It's not going to go very far, maybe half a meter away from the parent plant, and that's enough just to get out from the shade of the parent plant. The seeds won't grow immediately when they land. Okay, they go through what's known as a dormant stage. So in dormant, a dormant seed is not growing, and it's defined as a period of inactivity or very low metabolism when the seed doesn't grow. Nothing seems to be happening. Externally, nothing seems to be happening with the seed. And this period of inactivity can last for many months. And obviously, it's to allow the cold winter months to pass. So seeds are usually produced in autumn, and they're going to spend the winter on the ground, and they won't start to germinate until the spring. So during winter, it's too cold, there isn't enough sunshine for young leaves to photosynthesize. So the plant stops growing, it becomes dormant. So it allows the seed to wait until the cold, dark winter weather has passed. In particular, there isn't enough light during winter time for seeds to photosynthesize. It also allows time for the embryo to develop. The little embryo, the radicle and plumule inside the seed, is continuing to grow at a slow rate. Okay, so it ensures that when the embryo is fully formed, that dispersal would have happened. Okay, you're also required to know some methods that a gardener might use to speed up the germination process to try and skip dormancy. Okay, one way that gardeners can do is they can break dormancy by scratching the outside of a seed, because normally the seeds would have a very tough testa that will normally take months of weathering to be broken down, of frost and rain to slowly break them down. So one method is scratching the surface of the seed like that to break the testa to allow germination. Okay, just to mention here that dormancy is caused by a plant growth regulator called abscisic acid. So it's a chemical that the seed produces, which is an inhibiting chemical. It prevents germination. So again, it's going to take a couple of months for the abscisic acid to be broken down inside the plant, inside the seed. And when the abscisic acid is broken down, either by weathering or other enzyme action inside the seed, when the abscisic acid is broken down, that allows germination to happen. Now, once germination begins, what does that mean? Germination is the growth of an embryo after dormancy into a new plant. So as the seed begins to grow after a period of dormancy, or as the embryo begins to develop into a new plant. Remember that the embryo inside the seed has a radicle and a plumule. The radicle is going to become the new root, and the plumule has begun to become the new shoot. So this is the radicle and plumule in green here. And seeds also have a food store. Now, this food store could be in the form of a cotyledon, or it could be in the form of an endosperm. So the food store is going to be a store of starch inside the seed. And this is why we eat seeds as a food store, because of the starch that's in the seeds. Okay. Now, in order to access this stored starch inside the seed, enzymes are produced. And these enzymes, in particular amylase, will break the starch down into simple sugars. So we have a food store inside the seed that consists mostly of starch. Once germination happens, amylase is produced to break the starch down into sugars. These simple sugars can move across to the little embryo plant, the radicle and plumule, where they're going to be used in respiration. The sugars are used in respiration to produce the ATP, the energy that's needed for the seeds to germinate. So what are the conditions that the seeds need? Seeds need to have water, oxygen, and a suitable temperature. Okay, so most people get the water and the oxygen fine, and they might write something like heat or warmth, or just the word temperature. You have to say suitable temperature to germinate. So the suitable temperature provides the optimum conditions for the enzymes inside the seed to function. You might remember that enzymes are affected by temperature. Enzymes slow down at low temperatures. Okay, so we need to have a suitable temperature for these enzymes to begin to function. The enzymes like amylase that are needed to break down the starch. Water is needed to activate these enzymes. Okay, so water needs to soak into the seeds to activate the enzymes. The enzymes then begin to break down the starch into sugars, and the enzymes involved in respiration become active. Why does the starch need to be broken down? Okay, you need to produce soluble sugars, okay, that can move from one location in the seed over to the embryo, and the embryo needs to respire them. The sugars are used in respiration as the embryo grows. Oxygen is needed by the embryo for respiration to provide ATP needed for cell division and growth. So aerobic respiration is happening here. Okay, so these sugars, again, they've been, like in respiration, they've been broken down into pyruvic acid, and they're moving into the mitochondria of the embryo cells, where they get broken down to form ATP. The ATP is needed is needed for cell division, for mitosis, and also for growth, for the manufacture of new materials, new cells. If we're having a look at this little diagram here, it's from a past exam question. And you'll notice that it's measured in dry mass, is how heavy it will be if the water is removed. Okay, in terms of dry mass, the endosperm is getting lighter and lighter and lighter because the stored food is being converted to sugars and being respired. And the embryo is getting heavier. The embryo is gaining mass because it's growing. New cells are being made. And at this point, the dry mass really shoots up. So this is where photosynthesis begins to happen. The plumule has formed new leaves. Okay, so it's manufacturing energy, sugar molecules at a fast rate. Okay, a quick look at the stages of seedling growth. So in this picture here, we're looking at the radicle emerging first. That starts to absorb water and passes the water back, activates all the enzymes. Okay, the plumule emerging. Now, the energy required to make this little plumule come out comes from the stored food. Okay, so the stored food is the cotyledon in this case inside the seed, and that's withering as we're going along. Okay, enough energy to produce the stem and the leaves. And as soon as the leaves are formed, that's when photosynthesis can take over, okay, into production of sugars. So all it needs, the plant needs at this stage is water from the soil, carbon dioxide from the air, and some sunlight, and it can manufacture its own biomolecules. So the important thing, the radicle emerges first to form a root, and the plumule emerges to form a shoot. So because this is dry mass, initially there's a loss in mass because the stored food in the cotyledon or endosperm is being converted into sugars and being respired. So the food is being used up. It will start to get heavier then because the plant begins to photosynthesize, okay, and it's manufacturing new sugars. Now, there are two experiments at the end of this chapter. Okay, so we're going to have a look at the theory behind each one. First one is investigating the effect of water, oxygen, and temperature on germination. Okay, these are the three conditions that seeds require to germinate. So we're going to use cress seeds because they grow very quickly. You can have a result in a week. So for the control dish, place cress seeds onto wet cotton wool in a petri dish and leave in a warm area. This is the control. They have all the conditions needed to germinate. Water, because the cotton wool is wet, oxygen, and you leave them somewhere warm, suitable temperature. So in the photograph, this little dish here, wet cotton wool, seeds, and you leave them somewhere warm in the room. And within a week, they'll start to grow. They have all the conditions. Now we're making up our experimental. Leave another dish of seeds in the fridge. The temperature isn't suitable, so the seeds don't germinate. So these dishes are placed in their wet cotton wool in the fridge. They don't germinate. Leave another dish in an anaerobic jar, which is a large jar. We put a chemical inside the jar as well that removes oxygen from the air. Okay, these seeds have no oxygen and they don't germinate. So these seeds here are placed in an anaerobic jar. They have no oxygen. There's a chemical called alkaline pyrogallol added, or a similar chemical that's used to remove oxygen. And finally, if we put some seeds onto dry cotton wool, these seeds have no water. They have oxygen at a suitable temperature, but they have no water. So if they're dry cotton wool, they won't grow. So it's quite a simple experiment. It can be done in test tubes or it can be done in petri dishes like this. And the purpose for each of those factors, water is needed to activate the enzymes in the seed for germination. The warm temperature provides the optimum temperature for the enzymes to work. So water activates the enzymes. The warm temperature provides the optimum temperature for the enzymes to work. And oxygen is needed for respiration. The growing seeds need to respire. Oxygen is needed for that. And finally, this experiment is investigating the enzyme that is produced during germination. So we're using starch agar. Okay, we have a choice, so we're using starch agar to show digestive activity during germination. So the background to this, we've covered it already. Seeds produce the enzyme amylase to break down stored starch to simple sugars used in respiration to provide energy. So the seeds are making amylase. Amylase, remember, breaks down starch to produce simple sugars. We're going to use broad bean seeds because they produce a lot of amylase. Okay. We soak them overnight to activate the enzymes. They'll need water to start the whole germination process. And as a control, we're going to boil half of the seeds to kill them and denature the enzymes. So some of the seeds have been soaked as normal, the other seeds were soaked and then boiled. We're going to disinfect both sets of seeds so they can be dipped into Milton or some disinfectant because we don't want any mold or bacteria on the surface of the seeds because they'd quite happily grow in our dishes as well. Cut the seeds with a sterile blade, sterile to avoid contamination, to expose the enzymes, and place the seeds cut surface down onto starch agar. This jelly here is called starch agar. It just has a little bit of starch added to agar. Agar is a jelly. The starch agar provides a substrate for amylase enzymes produced by the germinating seeds. So these seeds are producing amylase, and some of the amylase will soak out into the agar. After 3 days, add iodine solution to the starch agar in each dish. So we're just pouring iodine solution onto each dish. The dish that contained the live germinating seed showed clear patches. Okay, you might remember that when you add iodine to starch, normally you get a black color. If there's no starch present, it will remain clear. So first of all, when iodine is poured onto this dish, we can see clear patches around all the seeds, a little bit of black, bluey black near the edge because there's a bit of starch still here. So what has happened? Amylase from the seeds has soaked into the agar, broken down the starch. So when I added the iodine, we get a negative result here. For the control, okay, these seeds were boiled. The amylase was denatured, so no more fresh amylase was made. The seeds were dead. So no amylase soaked into the starch agar, so the starch was not broken down, it's still there. So when I add iodine, we'll get that black color. Okay, so no digestion occurred in this dish. Okay.